Viruses, long seen as enemies, have been explored for their potential to treat cancer. This exploration began over a century ago with a surprising observation: certain viral infections seem to reduce cancer symptoms. This led to numerous experiments throughout the 20th century, yielding mixed results and significant challenges. However, the advent of genetic engineering in the 1990s provided new hope. By modifying viruses to specifically target and destroy cancer cells, researchers have made significant progress. Notably, the Vaccinia virus, once used to eradicate smallpox, has shown promise in treating various cancers. This script delves into the history, challenges, and breakthroughs in using viruses as a weapon against cancer, offering a glimpse into a potentially transformative approach to cancer treatment. In 1904, a remarkable event occurred that would pave the way for future cancer research. Dr. George Dock noticed that a leukemia patient's cancer improved significantly after the patient contracted influenza. This surprising observation suggested that viral infections might have the potential to fight cancer. Intrigued by this possibility, researchers began to explore this phenomenon further. By the 1950s, Dr. Chester Southam took this exploration to a new level by conducting more structured studies. He injected various viruses, including the common flu virus, into cancer patients. The initial results were encouraging, with many patients experiencing a reduction in tumor size. However, these early experiments also uncovered serious risks. Some patients developed severe viral symptoms, and unfortunately, a few succumbed to these complications. These tragic outcomes highlighted the dangers of using unmodified viruses for cancer treatment. Despite these setbacks, Dr. Southam's trials provided valuable insights and laid the groundwork for future research. They demonstrated that under the right conditions, viruses could potentially target and combat cancer cells. This early work set the stage for the development of safer and more effective virus-based cancer therapies. The 1990s saw a revolutionary shift in medical research with the advent of genetic engineering, opening new avenues for cancer treatment. Scientists began to explore how modifying the genetic structure of viruses could enhance their ability to fight cancer. This period marked the development of the first genetically modified vaccine for hepatitis B, a significant milestone in medical science. This breakthrough inspired researchers to apply similar genetic engineering techniques to create viruses that could specifically target cancer cells. By carefully altering viral genes, scientists aimed to ensure that these engineered viruses could distinguish between healthy cells and cancerous ones, attacking only the latter. This precision was crucial in reducing the risks associated with earlier virus-based treatments. Researchers focused on enhancing the specificity and efficiency of these modified viruses, leading to more targeted and less harmful treatments. The success of these early genetic modifications brought new hope to the field of oncology. It demonstrated the potential of using genetically engineered viruses as a viable and effective cancer treatment, significantly advancing the quest for safer and more targeted therapies. Building on earlier advancements, cancer biologist Frank McCormick and his team developed Onyx 015, a genetically modified adenovirus designed to target cancer cells specifically. This virus was engineered to exploit the weaknesses found in cancer cells while sparing healthy ones. The key innovation was its ability to target cells with deactivated p53 proteins, a common feature in many cancer cells. These proteins usually help regulate cell death, but when deactivated, they allow cancer cells to grow uncontrollably. Initial laboratory tests showed promising results, with Onyx 015 effectively killing cancer cells. However, the virus faced challenges, such as struggling to replicate efficiently and passing on its modifications to subsequent generations. Despite these hurdles, Onyx 015 demonstrated the potential of genetically engineered viruses in cancer therapy. This groundbreaking work laid the foundation for further developments in the field, highlighting the promise of using modified viruses to treat cancer. The researchers then focused on the Vaccinia virus, known for its role in eradicating smallpox. Given the widespread immunity from smallpox vaccinations, Vaccinia was considered a safer candidate for genetic modification. Scientists modified the virus to produce proteins that specifically target and kill cancer cells without triggering the immune system. This approach ensured that the virus could enter cancer cells, produce toxic proteins to destroy them, and cut off their blood supply, effectively starving the tumor. By targeting the cancer-specific MyC protein, the modified Vaccinia virus showed significant potential in preclinical trials. These trials indicated that the virus could precisely attack cancer cells while leaving healthy cells unharmed, marking a significant step forward in virus-based cancer therapy. following successful animal trials, the FDA approved the first human trials of the modified Vaccinia virus in 2022. Conducted by the City of Hope Hospital and the Australian company Imugene, these trials involved 100 patients with advanced solid tumors. To enhance safety, an additional drug, pembrolizumab, was administered alongside the virus. The results were promising, with reductions in tumor size observed in cancers such as pancreatic, lung, breast, and ovarian. Inspired by these findings, researchers are now preparing for larger phase II trials involving thousands of patients. These larger trials aim to confirm the virus's efficacy and safety on a broader scale. Parallel efforts in India, led by Tata Memorial Centre and IIT Bombay, are developing affordable cancer treatments using similar techniques, potentially benefiting a broader population. The Indian trials focus on making these advanced treatments accessible to more people by significantly reducing costs, thereby offering hope to patients who might otherwise not afford such therapies. The journey from early viral observations to advanced genetic engineering highlights the great potential of virus-based cancer treatments. These treatments offer new hope for more effective and accessible cancer therapies. Although challenges and risks remain, the progress made in recent decades has been promising. Ongoing research and human trials indicate that using viruses to cure cancer is becoming increasingly possible. The main advantage of this innovative approach is its ability to specifically target and destroy cancer cells while sparing healthy cells. However, there are also disadvantages, such as potential side effects and the complexity of modifying viruses. Despite these challenges, virus-based treatments could transform cancer care, providing new lifelines for patients worldwide. As research continues, the future of cancer treatment looks brighter, with the possibility of making cancer not just treatable, but curable. For more updates on groundbreaking research and health innovations, subscribe to our channel.